Hello, everyone, and welcome to today's webinar. I'm Charlene Lancaster, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speakers, Noor Gamo and Ravi Amravati, will discuss emerging research on autophagy's context-dependent roles in cancer and the challenges of targeting this pathway therapeutically. Before we begin, I'd like to take a moment to thank today's webinar sponsors, Taconic Biosciences, BioXL, Aviva, and Syncell. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point, and our speakers will address these during the Q&A sessions following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsors have provided us with some helpful resources related to today's webinar, and you can download these at any time under the handout section located on the right side of your screen. If you've minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our first speaker. Noor Gamo earned her PhD in Trieste, Italy, under the mentorship of Lawrence Banks, where she investigated the role of human papillomavirus-encoded proteins during oncogenesis. In 2009, she moved to Memorial Sloan Kettering Cancer Center for her postdoctoral training and began exploring the autophagy field. After completing her training in 2014, Gamo established her laboratory at the University of Edinburgh, where she focused on autophagy and brain tumor development, and holds a Cancer Research UK Career Development Fellowship. Let's make sure our slides are up and running. Okay, Noor, take it away.
Hello, it's my pleasure to be presenting today's webinar. My name is Noor and I'm based at the University of Edinburgh. And my lab is interested in studying autophagy and its role in aggressive brain cancers. So what is autophagy? In brief, autophagy is a cellular homeostatic pathway. It involves the degradation of cellular components. So during autophagy, we have these double membrane vesicles, also known as autophagosomes, that can recognize and encapsulate cellular components that are targeted for degradation. Autophagosomes, when mature, can fuse readily to the lysosomal system, which provides the enzymes required for the degradation of the autophagosome content. And we think autophagy has important functions in the cell. Primarily, this degradation of unwanted molecules is important to remove any toxic material in the cell, but also the recycling of nutrients back to the cell is important, especially during periods of nutrient starvation. So what is the molecular autophagy pathway? I will summarize it here briefly. At the heart of the pathway, there are these ubiquitin-like molecules, one of which is known as LC3. LC3 is usually inactive and sits in the cytosol. However, when autophagy is activated, it can be conjugated to a lipid moiety known as phosphatidylethaноlamine. This conjugated form of LC3, known as LC3-2, can be now tethered to the autophagosome membrane. Now, this conjugation reaction can be controlled by a number of upstream complexes, such as the ATG5 complex and the VPS34 complex. Further upstream, there are a number of signal molecules can act to regulate autophagy, such as mTORC1 and AMPK. This is important because both of these signaling pathways are involved in cancer, suggesting that autophagy can be highly regulated in cancer cells. So what happens during autophagy, specifically during the recognition of autophagic substrate? Surely we do not want to be degrading any cellular components that is required for the cell. So for the recognition of autophagic substrate, there exist a number of receptor proteins that can bind specifically to the autophagic substrate through, for example, eat me signals. These receptor proteins often would bind to autophagy proteins such as LC3, leading to the recruitment of the autophagic substrate to the autophagosomal membrane. So here, for example, we have a scenario where a substrate can be modified by a ubiquitin chain. This modified substrate can be recognized by a receptor protein such as P62. P62 can bind to LC3, leading to the recruitment of the substrate to the autophagosome. So many labs have been interested in studying the role of autophagy in cancer, and this is what we currently think autophagy plays in terms of cancer progression. It's thought that at early stages of tumor formation, when a normal cell is undergoing cellular transformation, autophagy acts to inhibit this tumorigenesis initiation. And this is thought because of the role of autophagy in maintaining cellular homeostasis, which is important to maintain DNA integrity. On the other hand, when tumors are formed and well established, it's thought that autophagy is required in this stage. And this is for a number of reasons, depending on the tumor type, but generally involves the requirement for autophagy to provide the metabolic needs of cells and also to regulate inflammatory signals. But we all agree that it's important to study the role of autophagy in a context-dependent manner, meaning that we need to factor in mind the tumor type, the oncogenes involved, and the tumor stage. And this is just an image from a review summarizing the multiple ways by which autophagy has been shown to be involved in tumor formation and progression. As I mentioned, my lab is interested in studying the role of autophagy in an aggressive brain tumor known as glioblastoma. Glioblastoma is the most common and lethal type of brain cancer. And many studies have been performed to analyze the oncogenes and the tumor suppressors that are mutated in glioblastoma. And these studies have identified mutations in EGFR and PGFRA as commonly mutated to drive glioblastoma progression. So when we were first thinking of the question, does autophagy play a role in glioblastoma formation, we asked a very simple question. Can autophagy regulate the oncogenic signaling of these receptor tyrosine kinases that are mutated in GBM? So to start with, we wanted to analyze the receptor trafficking and signaling, and to do that, we basically treated cells with a growth factor relevant that binds the receptor tyrosine kinase. For example, it would be EGF in terms of EGFR. Activation of the receptor tyrosine kinase leads to its endocytosis and signal activation, and subsequently the receptor would often take two different routes. The receptor would either be recycled back to the plasma membrane, which is important to maintain its signaling, or it could be targeted to late endosomes and lysosomes for signal termination. So to start with, we asked the question, does autophagy regulate EGFR? And to do this, we treated cells with EGF and looked at signal activation. And using cells that we could engineer to be inhibited of autophagy, so here we're targeting ATG7, which is a core autophagy protein, we can see that when cells were treated with EGF, there was a strong suppression of EGF-mediated signaling in terms of AKT and ERK phosphorylation, suggesting that in the absence of autophagy, EGF signaling is strongly suppressed. Now we could show this in another way. We looked at the ability of growth factors to mediate cell survival. So in the absence of growth factors, cell death can be induced. And we can monitor this by propydium iodide staining in cells. So in control cells, if we remove growth factors, cell death will be induced, as you can see here by propydium iodide positive cells. However, if we add EGF to the cells, this induction of cell death can be suppressed and lead to cell survival. However, in autophagy inhibited cells, cell death is also induced in the absence of growth factor. However, the cells do not respond to EGF's treatment, suggesting that indeed autophagy inhibited cells have defective EGFR signaling. So what's happening to EGFR? Why is it suppressed in the absence of autophagy? So to do this, we followed receptor trafficking in cells. First, we could show that in the absence of autophagy, the receptor can be targeted to late endosomes normally. However, what was interesting is that when we monitored receptor recycling to the plasma membrane, we could see that it was suppressed. And in fact, the receptor was accumulating at early endosomes. And I'll just show you briefly the data to support this. So we can see here when we co-localize EGFR with a recycling endosome marker, RAB11, there's a reduction in the ability of EGFR to go to these recycling endosomes in the absence of autophagy. And this is supported by the fact that the receptor cannot be recycled back to the cell in the absence of autophagy, which is, as I said before, is important to maintain signal activity. What we observed was that EGFR accumulates at these early endosomes. But what's interesting is that these early endosomes also stain positive for galectin 8, which is a marker of perturbed endomembranes. So in the absence of autophagy, early endosomes appear to stain more positively to galectin-8. This suggested to us that perhaps autophagy can recognize perturbed early endosomes. So when autophagy is inhibited, these perturbed early endosomes can accumulate. So to look at this, we stained GFP-LC3 expressing cells with EEA1, which is the marker of early endosomes. And we can see that there's a slight co-localization between early endosomes and autophagosomes in the absence of any treatment. And this can be further increased if we treated cells with baflomycin to inhibit lysosomal degradation. So we thought, can we induce this co-localization further by perturbing early endosomes? So we treated cells with monensin, which is a known reagent that can perturb endocytic vesicles in cells. And indeed, we can see that the co-localization between EA1 and LC3 is robustly enhanced in the presence of monensin, suggesting that indeed autophagosomes can recognize perturbed early endosomes in cells. Does this lead to the recruitment of lysosomes? So to look at this, we co-localized the lysosomal marker LAMP2 with the EA1 as a marker for early endosomes. And we can see again here that LAMP2 can recognize the early endosomes in the presence of monensin. Is the autophagy machinery required? So to do that, we looked at this co-localization in the absence of ATG7 to inhibit autophagy. And as you can see here, in the absence of ATG7, the ability of lysosomes to be recruited to early endosomes in the presence of a stress is inhibited. So this suggested to us that indeed autophagosomes are involved in recruiting lysosomes to perturbed early endosomes. So what we think happens in this scenario, EGFR normally is recycled back into the plasma membrane, which is important for signal maintenance. However, when autophagy is inhibited, it appears that EGFR can be arrested at these subset of perturbed early endosomes, leading to its inhibited recycling and inhibited signaling as well. So this is great in terms of EGFR, but is the same regulation happening in terms of PGFRA? Does autophagy regulate PGFRA similar to EGFR? From what we know from the literature, we suspected that if there is a regulation, it will occur in a different mechanism, mainly because we know that EGFR, while EGFR is recycled in the cell, PDGFRA is not recycled. Instead, PGFRA is often always targeted to late endosomes. However, it can still signal from late endosome, which is unlikely, unlike other receptor tyrosine kinases, which were thought that late endosomes act as signal suppressors. So to look at the regulation of PGFRA by autophagy, we stimulated cells with PGFAA to stimulate PGFRA and looked at signal activation. And again, here we can see that in the absence of autophagy, here we're inhibiting autophagy by RNA interference. We can see that there's a suppression of PGFAA-mediated signaling activity. What was interesting is that we also saw that there is an increase in PGFRA level when autophagy is inhibited. So we wanted to chase what is the underlying molecular mechanism for regulating PGFRA by autophagy. We could confirm this increase in PGFRA level in the absence of autophagy by using small molecule inhibitors. Here we're targeting ULK1, which is a kinase involved in inducing autophagy. And you can see here inhibiting ULK by two different inhibitors leads to an increase in PGFRA level. On the contrary, if we induce autophagy by amino acid starvation, we can reduce the levels of PGFRA. Again, we use genetic inhibition against two different ATG proteins that are important for autophagy, ATG5 and ATG7, and we can see similarly an increase in PGFRA levels. So we reason perhaps since autophagy is a degradation pathway, could PGFRA be degraded by autophagy? So initially we looked at the ability of autophagy's essential proteins to co-localize with PGFRA, and we can see that both ATG16L1, which is an essential autophagy protein, can co-localize with PGFRA in cells. And in addition, LC3 and PGFRA can co-localize as well, and this co-localization can be enhanced by the addition of the lysosomal inhibitor baflomycin A1. So then we ask the question, well, if PGFRA is degraded by autophagy, then what is the receptor that mediates its recruitment to the autophagosome? And we did a very simple approach to tackle this. We use reagents available in the lab, and we thought to start by just asking whether P62 would be a potential receptor, which I have mentioned before is commonly looked at as a receptor in the autophagy pathway. First, we looked at the co-localization of PGFR to P62, and we can see that indeed both proteins can co-localize in the cells, and this co-localization can be further increased if we inhibit baflomycin or lysosomes with baflomycin A1, as we saw with LC3. What was interesting is that the knockdown of P62 is sufficient to enhance PGFRA levels, suggesting that indeed it could be the receptor involved in recruiting PGFRA to autophagosomes. And finally, we can observe co-immunoprecipitation between endogenous P62 and endogenous PGFRA in cells, and this co-immunoprecipitation can be enhanced by activating PGFRA with PGFAA and with inhibiting lysosomal degradation. Altogether, reinforcing the idea that P62 can be the receptor for PGFRA. So this is all great, but however, we do not explain why there is a reduction in signaling activity when autophagy is inhibited. So since there is more PGFRA in cells in the absence of autophagy, one would expect more signaling and not less signaling. So what is the reason underlying this inhibited signaling activity in the absence of autophagy? So to address this, we did similarly to what we did with EGFR. We looked at the trafficking of PGFRA in cells, and we observed that the ability of PGFRA to localize to early endosome is not inhibited in the absence of autophagy, occurs normally. But what was interesting is that the ability of PGFRA to go to late endosomes was actually suppressed in the absence of autophagy. So here we can see that the co-localization of PGFRA with the late endosome marker RAP7 can be reduced in the absence of autophagy. Now this is important. If you remember, I said before that unlike other receptors, PGFRA can still signal from late endosomes. So can we recapitulate what we observe with autophagy inhibition if we just simply inhibit RAP7? And that's indeed what we saw. So if we inhibited late endosome trafficking with RAP7 knockdown, we can also see an increase in PGFRA levels as well as a decrease in downstream signaling, suggesting that indeed this trafficking to late endosome is important for receptor trafficking and signaling. So what we think happens in terms of PGFRA, the receptor usually would go to early endosome and late endosome when activated, which is important for its signaling activity. However, in the absence of autophagy, we see a disruption in the ability of PGFRA to go to late endosomes, which is important for its optimal signaling activity. So this is all great, but we would like to see whether this is relevant in terms of cancer progression. So to look at this, first we looked at patient-derived cells, glioblastoma cells, and we are fortunate in Edinburgh to have access to multiple different cell lines that are derived from patients, from glioblastoma patients specifically. And we requested 2 different cell lines that were known to have overexpression of PGFRA. And we can see that both these cell lines respond to autophagy inhibition. Again, here we have the ULK autophagy regulator inhibition. And we can see that PGFRA can be upregulated when autophagy is inhibited with these inhibitors, suggesting that in the patient cells, we can regulate PGFRA by autophagy. So is this relevant in terms of tumor formation? To do this, we looked at mouse models. And the first model we looked at is based on these ARCAS vectors, which was developed by Eric Holland. ARCAS vectors can be engineered to overexpress an oncogene of interest and simultaneously to express an shRNA targeting a pathway of interest. These ARCAS vectors or viruses that are packaged with them can be injected to mice brains that express the specific receptor for these viruses, and this will lead to tumor development with time. So we've engineered these vectors to overexpress PGFA, as well as express shRNA targeting ATG7 and ATG3 to inhibit autophagy. And we can confirm this system works well in culture cells. So you can see here an overexpression of PGFA, as well as a robust knockdown of autophagy proteins. When we looked at tumor formation, in the control cells, or the control setting where we have ARCAS PGFA expressed along with shRNA targeting a control sequence, so LAC-Z in this case, we can see that tumor formation happens as expected. However, when autophagy is inhibited by ATG3 and ATG7 shRNA, we can see a strong reduction in tumor formation. So based on our cell-based study, that this suppression of tumor growth is due to the ability of autophagy to support PGFR signaling in cells. So is this the case? So to address this question, we thought that if we hyperactivate downstream signaling, for example, by PTEN deletion, which leads to hyperactivation of AKT signaling, then we no longer would need autophagy for tumor growth. Fortunately, we knew that our mouse models has a flux version of PTEN, which means that if we co-inject ARCAS vectors that express the Cre recombinase, we can delete PTEN in our system. So we again tested our system in culture cells, and we can see that indeed we can delete PTEN when expressing the Cre recombinase, and similarly still get robust knockdown of our autophagy genes. And now when we looked at tumor formations in animals, we see that whether autophagy is intact or inhibited, tumors can form when PTEN is deleted, suggesting that in the absence of PTEN, tumors no longer rely on autophagy because signaling is being hyperactivated. Many of you would agree there's no single perfect mouse model to study tumor formation, so we also made use of another mouse model that we have access to in Edinburgh. This is based on a model developed by Steve Pollard in Edinburgh, which relies on the injection of glioma stem cells into mouse brains. These glioma stem cells can be engineered to overexpress PGFR and have p53 inhibited. We further engineered them to delete PTEN. And again, using techniques from Steve Pollard's lab, we can get robust genetic editing in these cells, and can delete ATG7. So here we can have two different single clones that are deleted of ATG7, and each of them have a different sequence targeting ATG7 for CRISPR editing. So what happens in mice? When we injected these glioma stem cells that have intact PTEN, we can see that tumor formation happens robustly and as expected. However, when autophagy genes are inhibited, both single clones that lack autophagy are disrupted in their ability to grow in mice. So what happens in cells that are deleted of PTEN? Similar to what we observed before, in PTEN deleted cells, they no longer rely on autophagy for growth. So here we have similar growth of control and autophagy inhibited cells, suggesting that autophagy is specifically required for PGFR growth when PTEN is intact. So altogether, to summarize this part, our data show that autophagy is required for oncogenic signaling of EGFR and PGFRA. This is through the ability of autophagy to regulate receptor trafficking. And we show that PGFRA-induced tumor formation require autophagy, but only when PTEN is intact. So this is all great, but we would like to think of this more in terms of therapy. Can we regulate autophagy more chemically in order to suppress tumor growth potentially in the future in humans? As many of you would know, there are no specific inhibitors for autophagy. However, we are currently looking at two different approaches. We would like to inhibit early stages of autophagy by targeting metabolic regulators. I will not discuss this today. However, what I would like to discuss what we've been currently doing to target autophagy at early stages, late stages through lysosomal degradation. So one of the approaches we took was to screen for small molecules that will suppress glioma stem cells growth and inhibit autophagy. And through this screen, we identified a group of molecules that also had antipsychotic effects in humans. This is interesting for us because in terms of brain tumor treatment, we require the ability of small molecules to penetrate the blood-brain barrier. So these molecules are known to cross into the brain. The interesting things about some of these molecules is that they are able to suppress glioblastoma cell growth, but not, to a lesser extent, to suppress normal cell growth. So we wanted to take these inhibitors further and understand how they work. We observed that these inhibitors can suppress autophagy at late stages. So you can see here that treatment with these molecules will accumulate LC32, which is the lipidated form that goes to autophagosomes. And because there's no further increase when lysosomes are inhibited, with baflomycin, this shows us that these molecules suppress the lysosomal degradation of SU32. We further show that these molecules can go to lysosomes. They accumulate in lysosomes by co-localizing them with lysosomes, so we can visualize them in cells. And further, we looked at lysosomal acidification using lysotracker, which fluoresces in acidic compartments. And we can see, similar to baflomycin A1, which is a known inhibitor of lysosomal pH, our molecules of interest can also suppress lysosomal pH, suggesting that they act by suppressing lysosomal degradation. What's further interesting to us is that if we looked at two structurally related molecules, so these molecules are known to accumulate in lysosomes or predicted to accumulate in lysosomes, we do not see the similar inhibition of lysosomal degradation. So these molecules do not appear to inhibit SU32 degradation in cells. And this is also correlated with their inability to suppress cell growth. So unlike our molecules of interest, treating cells with these structure-related molecules do not suppress cell growth. So would our identified antipsychotic inhibitors be relevant in suppressing tumor growth in mice? To start to look at this, so these are all preliminary data that we're currently working on. We starting to look at glioma stem cells that are injected into mice brains and following tumor formation in settings where there's no treatment or treatment with the antipsychotic molecules. And we're starting to see that indeed our molecules of interest can suppress tumor growth in animals. So what we're currently doing, we would like to understand better how these molecules inhibit lysosomal function and why do the cells die? Why do they die in response to lysosomal inhibition? Hopefully this will help us understand better how we can target glioblastoma in a more therapeutic manner. So with this, I would like to end and thank people in the lab who've done this study, and also our collaborator within the University of Edinburgh and Outwith. And I will be happy to take any questions at the end of this webinar.
Thank you for that presentation, Noor. We have several questions from the audience, so let's get to them. If you want to turn on your camera and your mic. Perfect. Okay, so what cancers showed promising responses to autophagy inhibition in the past?
Right. Okay. A lot of these studies have been performed in labs, so there aren't any convincing studies in the clinic, I don't think, or at least I'm not aware of. But one recent interest is the response of pancreatic cancers to autophagy inhibition. That seems to be involved in how autophagy can modulate immune response in pancreatic cancer cells. So, so out of many cancers, sounds like pancreatic cancer is responding well to autophagy or lysosomal inhibition, at least.
Wonderful. And can resistance from inhibiting autophagy develop during anti-cancer treatment?
That would be a big question to kind of look for. As with many other cancer therapeutics, at least, resistance is often occurs frequently, especially with aggressive cancers with multiple genetic modifications. It's highly possible that cancer cells will develop resistance to autophagy inhibition or lysosomal inhibition, and trying to understand mechanisms how they can do that would be quite important, because these are all vesicular trafficking pathways and cells can modulate them in different ways in order to bypass the chemotherapeutic effects.
Understood. And what are the main challenges of developing specific autophagy inhibitors?
Right. This is when I first started working in autophagy, this was a dream for me, is to look for autophagy inhibitors. But I think now we've not seen many specific inhibitors, mainly because of the kind of enzymatic reactions that take place during autophagy. There has been some kinase inhibitors that were developed against the ULK complex, which is an upstream complex that regulates the initiation of autophagy, but they seem to have some non-specific effects on cells, which is common for kinase inhibitors. But it would be interesting to see how they develop further. But yeah, I imagine it's to do with the kind of the ubiquitin-like activities of the autophagy central enzymes that would seem to be hard to target.
And your new your lysosomal inhibitor, are you expecting it to have effects in other cell types in the body?
So in terms of toxicity,
Yeah.
that would be interesting to look at. So the agents that we're looking at have been used as antipsychotic agents. So I think it will all depend on the required dosing, so the concentration of the drug that needs to be used, the number of times it's administered. So yeah, that would be interesting. We know from genetic studies that inhibiting autophagy at least will have eventual toxic effects in animals. But whether that will translate similarly in terms of small molecules would be interesting to look at. Yeah, so I think there will be a number of factors that will be important to look at to determine what's the overall effect on the human body when we target lysosomal degradation.
Yes, because if it affects other cell types, of course, that'll affect like the immune system, and then you have to worry about even just regular housekeeping, even trying to get rid of old cells. And so it'd be really interesting if you could figure out maybe an inhibitor that could only target specific aspects and not others.
Yeah, I mean that's always the biggest challenge in cancer treatment, isn't it? And I mean, I always say, the most commonly used anti-cancer agents, they target DNA replication and will have toxic effects. So I think it will be about dosing, so the concentration that is going to be used, how frequently, and also the kind of assessing the benefits versus the adversaries of the negative effects of the drugs that we're using. So I think there's definitely lots of things to keep in mind, and the best way to know would be to conduct the trials once we're confident with any small molecules that we're developing.
And is there any kind of computational models that you could use to help kind of maybe you're simulating receptor interactions or something like that? Moving forward?
So yeah, I mean, we can think of computational models in terms of drug development, maybe. So in terms of looking for small molecules that will be predicted to cross the blood-brain barrier, for example. I can. That's the first thing that occurs to my mind, but I'll probably will need to think about this question more. Yeah, in terms of how computational models. My lab is definitely not a computational lab, so it's not something we do frequently. But yeah, that's the first thing I can think of. I know our chemist colleagues, they have started to look at sort of structures of molecules and how can we shortlist drugs to test in terms of brain tumor targeting.
Fantastic. Well, thank you, Noor, for that discussion. We really appreciate your presence here today.
All right, thank you very much for the opportunity.
If anyone has further questions, please consider reaching out to the speaker directly. Her contact is shown on the screen. So now let me introduce our final speaker. Ravi Amravati is a physician scientist focused on translational research related to cancer therapeutics. He was among the first scientists to demonstrate that autophagy is a target for cancer therapy using animal models. Collaborating with many clinical investigators, Amravati has translated these findings into phase 1 and 2 clinical trials. He has also developed novel and potent autophagy inhibitors and identified PPT1 as a new target in the autophagy pathway. So let's make sure our slides are up and running. Okay, if you want to start presenting, that's good. Okay.
All right. Thank you so much to the scientists and the organizers for this opportunity to present our work. I'm Ravi Amravati. I'm a physician scientist at University of Pennsylvania. And today I'm going to talk to you about our latest paper on targeting autophagy in cancer, acute and chronic effects of PPT1 inhibition in melanoma tumorigenesis. Here are my disclosures. And you heard from Dr. Gameau about the autophagy pathway, so thank you to her for introducing this. But the key takeaways of this talk are shown here. The autophagy and lysosome pathways promote growth and survival of cancer cells, making them attractive targets for cancer therapy. We focused on an enzyme called PPT1, or palmitoyl protein thioesterase 1, and it's a lysosomal enzyme that regulates autophagy. But what we observed most recently was that if you block PPT1 activity using chemical inhibition, siRNA, or CRISPR knockout, you actually get vastly different proteomic changes, effects on ferroptosis and anti-tumor activity. We then took this in vivo with a conditional knockout mouse model and found that if we knock out PPT1 in a genetically engineered mouse model of melanoma, we found no impact on tumor initiation or survival of mice. So the conclusion of this talk is really that genetic PPT1 inhibition produces different adaptations than chemical PPT1 inhibitors, and we need to study the mechanism of chloroquine derivatives further beyond PPT1 in cancer. So let's get some background out of the way. And you heard about the autophagy pathway from Dr. Gameau. It's shown on the right. Just to recap, this is the cell's way of sequestering organelles like mitochondria, shown in brown, and forming autophagosomes that then are trafficked to the lysosome, and the cargo is degraded and, importantly, recycled. And it's that recycling capacity and the removal of damaged organelles that are the key for autophagy's survival mechanism in cancer. And for that reason, and for some of the reasons that Dr. Gameau mentioned about the difficulty in targeting enzymes in the upstream aspects of autophagy, we have focused our efforts on targeting the lysosome. It turns out that the metabolic stress of the tumor microenvironment and most cancer therapeutics will upregulate autophagy in cancer, and that's why it's been an attractive target. When we focused on targeting the lysosome to block this pathway, we started with chloroquine derivatives as a known lysosomal inhibitor. At the time, chloroquine had no molecular target, and I'm going to hopefully convince you that PPT1 is a molecular target of chloroquine derivatives. This is an enzyme that depalmitylates proteins and regulates lysosomal acidification. When you block PPT1, you definitely block autophagy. So it is a very potent autophagy inhibitor. And interestingly, there are humans that are born with mutations in PPT1 who lack PPT1 enzymatic activity throughout their body. And these children are born normal. You can't tell that they have any disease. But as time goes by, as years go by, they develop CNS disease, starting with retinopathy, which turns out to be the main side effect for chloroquine. So this human condition of PPT1 deficiency is a telltale sign that this could have a therapeutic window in cancer, where we're talking about treatments that don't need to last for years. And on that note, our group has been instrumental in launching hydroxychloroquine clinical trials. And we've conducted over a dozen or so clinical trials in various cancers with really mixed results. In some cancers, such as melanoma and pancreatic cancer, we've seen increased response rates, but some of the durations of response are not that long. So this suggests that there may be potency issues and or resistance mechanisms, as was brought up in the question and answer session. And we've been working on both of those. Today, I'm going to be talking mostly about the potency issues, where we've developed a series of novel chloroquine derivatives that target PPT1. And you can see some of the references here and this review article, which is the more up-to-date review article on this topic. So the starting point for our novel chloroquine derivatives, if you look at the top left, you see the structure of chloroquine. The ring structure is the aminoquinoline motif. And you can see there are some nitrogens here in the tail. And hydroxychloroquine, the only difference is this hydroxyl group. So we worked with Jeff Winkler, a chemist at University of Pennsylvania, and he suggested dimerizing this molecule, which had been done in malaria and had never seen the clinic, although it looked very much more potent in malaria, the dimer form versus the monomeric form of chloroquine. So we came up with a novel dimer called lyso-1, and it has 2 aminoquinoline rings and a triamine linker with the central nitrogen that's methylated. And although there's only 2 ring structures here, lyso-1 is 10-fold more potent than hydroxychloroquine or chloroquine and has single-agent anti-tumor activity at many different doses. It accumulates in the lysosome. The next step was to take that lyso-1 structure and expand the linker. And so with the longer linker, we reached a compound called DC661 that is 10 to 100-fold more potent than hydroxychloroquine. It's really, DC661 is 100-fold more potent. And we then tested the effects of switching out the heterocycle. Instead of a quinoline ring, we would do a quinacrine or acridine ring here. And those are dimeric quinacrines. Now, these are now 1,000-fold more potent than hydroxychloroquine. We also tested the effect of modifying this R4 motif with various chemical motifs, and really the ones that we're going to focus on today are the methyl group and the hydrogen group here. So with the most potent molecule, DQ661, that's a quinacrine, we tested the effects of DQ661 versus its analog, DQ660. The only difference between these two is that central nitrogen is methylated. They have the same exact pKa. Amazingly, the 660 localizes to the nucleus and causes DNA damage. 661 localizes to the lysosome, no DNA damage. They have opposite effects on autophagy, with the 660 inducing autophagy, the 661 blocking autophagy. And 661's mechanism of action is that it binds and inhibits PPT1. The reason why this leads to lysosomal deacidification is that PPT1 regulates the palmitylation and localization of the key components of the vacuolar ATPase. When PPT1 is blocked, the vacuolar ATPase is localized in the plasma membrane, not the lysosomal membrane. And so the lysosome is deacidified. So these compounds, even though they are weak bases, the reason why they're deacidifying the lysosome is not their inherent chemical structure, but because they are blocking a specific enzyme, PPT1. And the way that we found PPT1 is shown here. We use chemical biology tools. We took the warhead of DC661 and linked it to a benzophenone affinity label and then a desthiobiotine handle. So we fed this entire large molecule to cancer cells. It localized to the lysosome. And when we shine UV light on the cells, the protein that is bound to this warhead will be conjugated. And then we can pull down this entire protein chemical complex using the desthiobiotine and streptavidin columns. We can see specific pull down of PPT1 here, because if we add in free drug of DQ661, we can compete out this probe and block PPT1 pull down. So we did this with DQ661. We then did it with DC661. This is the chloroquine rings here. And we pulled down even more PPT1, and we're able to completely prevent it from being pulled down by free drug with competition. We did this for the lyso-5 molecule, or lyso-1. It's the same thing. And then we even did it for chloroquine itself, really demonstrating for the first time the molecular target of this drug that has been used for decades in humans without a molecular target. So what is this enzyme PPT1? Well, the mRNA expression levels are elevated in most cancer cell lines. In the TCGA database, the mRNA of PPT1 is increased in tumor versus normal. and high expression of PPT1 is associated with worse outcomes in a number of cancers. There is a company that's developing a novel PPT1 inhibitor for clinical trials, and I'll tell you more about that. So here are some in vivo examples of how chemical PPT1 inhibitors have significant anti-tumor activity. Our initial compound lyso-5 was shared with a number of groups. This is a group from MD Anderson, combining lyso-5 with the CDK4 inhibitor palbociclib in breast cancer. And this breast cancer xenograft model, there is single agent activity of lyso-5 that's modest, but when you combine it with the CDK4 inhibitor, you get almost 100% tumor growth inhibition. If you look at DQ661, which is more toxic and more difficult to dose in animals, when you combine with chemotherapy, you get an augmentation of chemotherapy in pancreatic cancer. But the most interesting one here is DC661, which has significant single agent anti-tumor activity. And we've, again, shared this molecule with these molecules with many groups. They published independently. They're now all commercially available for testing and are widely used as autophagy inhibitors. And Genoscience Pharma has also found a novel monomeric chloroquine derivative that is being tested in clinical trials. Now, this is a group from France that took our molecules and used computational biology modeling to show the structure of PPT1 in green and how and where these molecules might fit in and bind to this enzyme. And what they concluded was this is the hydrophobic groove where the palmitate would sit in. And you can see how nicely these molecules fit into that hydrophobic groove of PPT1. So with that background, I'm going to turn to the paper we published this year. Marianne Chrissy in my lab is the first author. It's published in the journal Autophagy in 2025, and really stems from this observation that with our chemical inhibitors, we were seeing a different effect when we knocked out PPT1 with CRISPR-Cas9. And so on the top left, you can see siRNA in melanoma cell lines, siRNA against PPT1. We have a PPT1 enzyme activity assay. We see suppression of PPT1 enzyme activity with siPPT1, and we see a 50% reduction in cell viability with siPPT1. With chemical inhibition with DC661, again, we see suppression of enzyme activity, and we see almost 100% cell viability suppression. But when we start with CRISPR knockout, again, we see complete loss of PPT1 enzymatic activity when you knocked out PPT1, but the cells are fine. They're able to survive this specific form of PPT1 inhibition. And we then developed an inducible CRISPR line where with doxycycline, over the course of 3 to 4 days, the cells lose PPT1. And this is the 5th day. You can see the PPT1 enzymatic activity is low, but the cells are okay. So what's going on here? We work with Xin-Yao Tang and Lin Beer at the Wistar Institute, who run the mass spec core. And we perform label-free proteomics on the 3 different ways to block PPT1. On the top left is the CRISPR knockout. On the top right is DC661, and then the siPPT1 shown here. And you can see that surprisingly, 3 different ways of blocking the same enzyme only produce 6 proteins in common that were significantly upregulated. And even in the PPT1 knockout versus siPPT1, we see only 42 proteins that are common. And you can see those proteins here. And there's a lot more to dive into this, but I wonder how common this finding would be if we went and did this with other targets in cancer with inhibitors CRISPR and SIPPT1. And if you looked at the pathways that were different amongst the knockout, the siRNA, you can see that the ferroptosis pathway is downregulated in this knockout condition. And if you compare the siRNA to the knockout, we are seeing an upregulation of ferroptosis in the siRNA. Now, ferroptosis has come up before in our research on DC661. What we found is the way that DC661 induces cell death is as follows. This was published in JCI in 2023. And DC661 activates lysosomal lipid peroxidation. And these peroxidized lipids weaken the membrane, leading to lysosomal membrane permeabilization, LMP, which in turn activates a series, or really almost all of the mechanisms of cell death that are well characterized, including ferroptosis. And so we looked at ferroptosis inducer RSL3 in our knockout condition as compared to the knockdown condition. And we saw drastically different effects that may explain why the knockout cells are doing just fine with PPT1 knocked out, because they are not really inducing ferroptosis. When you have this very prominent ferroptosis inducer, RSL3, the control is doing so, but the knockout cells are not really sensitive to ferroptosis induction, whereas knockdown PPT1 with siRNA, the acute knockdown, we see this sensitivity to ferroptosis induction. Now, that's all in vitro. Could there be a different effect in vivo? And to understand this, we actually had to do the hard work of creating a conditional PPT1 knockout mouse model. And we had a tough time doing this, so we actually pursued two different CRISPR strategies. One is to introduce the 5' and 3' LOXP sites around the PPT1 exon 1. We were aiming for simultaneously, but all we got was the 3', and we were able to make homozygous 3' LOXP mice. We then use in vitro fertilization with the Cas9 plus the 5' LOXP guide RNA, and we're able to get line 6. And this took a few years, actually. In the meanwhile, a new technique called Easy CRISPR, which is probably used by most people now, is that the guide RNA template includes both the LOXP sites and the exon 1. And this generated relatively quickly to line 25. And you can see here, if we take genomic DNA from mice from these manipulations and then expose them to Cre recombinase, we get the bands of interest that we're looking for in line 6 and line 25. Now, importantly, maybe most importantly, this conditional knockout mouse is now being used to study Alzheimer's disease and other neurodegenerative diseases at Harvard, given that the lysosome is such an important part of neurodegenerative disease. And the obvious point is that this would be very useful to study lysosomal storage disorder, especially organ-specific effects of lysosomal inhibition. And if you are interested in this mouse model, it is available. We've donated this to Jackson Labs, and it is available commercially. So to understand the role of PPT1 in tumorigenesis, we took both these line 6 and line 25 and crossed it to the Bosenberg genetically engineered mouse model of melanoma, which is driven by constitutively activated BRAF and a loss of PTEN. So this is a quite complex mouse model where there is expression of Cre-ER driven off of a tyrosinase promoter. So only in the melanocytes will this Cre-ER be expressed. the CRE-ER will be localized to the cytoplasm unless it encounters 4-hydroxytamoxifen. And if that is painted on the skin of the mouse, the melanocytes will express CRE-ER, and the CRE-ER will localize to the nucleus, where it can then activate this allele, the BRAF constitutively active mutated allele, and the FLOX P10, so P10 is lost. So the end result of painting 4-hydroxytamoxifen on the skin of these mice is a melanoma tumor that's shown here. You can see the pigment here, and the histology looks very much like a melanoma. And so this has been a workhorse for melanoma researchers, albeit a very difficult one with all the alleles here. And but we were able to cross this to get PPT1 knockout in this model. And the results were not according to our hypothesis, that we found that PPT1 is dispensable for tumorigenesis. And you can see here PPT1 wild type versus the knockout. They still have tumors. The tumors are the same size, and the mice still die of melanoma. Whether you have a floxed PPT1 or heterozygous or the wild type, there's no difference in survival. There was no difference between line 6 or 25, and no difference in males or females. And no difference in histology. You can see that the melanin pigment is produced extensively in this, and the cells are similarly abnormal. We wanted to know if our cell line effects on ferroptosis in the human cell lines also applied in the mouse cell line, possibly to this mouse model. So we took a cell line derived from this mouse model and knocked out PPT1 using CRISPR. Again, you see no PPT1 enzymatic activity, but the cells are able to proliferate fine, and they don't respond to the ferroptosis inducer. So our conclusion is that, one, chloroquine derivatives inhibit PPT1, but likely have other effects on lysosomal membranes that genetic inhibition does not reproduce. And our speculation is that we are activating an antioxidant pathway that may be upregulated during chronic PPT1 inhibition. And this is important because the PPT1 inhibitors are still moving through clinical trials. Here is GNS 561, the Genoscience Pharma, which is being tested in hepatocellular carcinoma with atezolizumab, and in liver tumors in combination with TACE, which is local injection of chemotherapy. In cholangiocarcinoma, a really unmet need, there's a single agent and combination phase 2 study that's ongoing in the US and EU. And, the FDA has granted orphan drug designation for this molecule. But really, hydroxychloroquine has made a major comeback with this paper. This is coming out of the Penn group, breast cancer group, and I was not part of this trial. Hydroxychloroquine was used in breast cancer patients who had undergone surgery, undergone adjuvant therapy, undergone tamoxifen therapy, all the possible treatments, but still had disseminated tumor cells in their bone marrow, still had breast cancer cells detectable in their bone marrow after all of the approved adjuvant therapies. There's actually a large number of these women around, and this trial accrued very rapidly. Patients with disseminated tumor cells after adjuvant therapy were randomized to hydroxychloroquine, an mTOR inhibitor, everolimus, or the combination, where we're inducing autophagy and blocking it at the same time, and compared to observation. This was a short course of drug therapy. And what you can see here is this is after the drug therapy or pre-treatment. There's a high level of disseminated tumor cells, and hydroxychloroquine treatment was really the most effective here. at producing nearly more than an 80% reduction, close to 90% here, of disseminated tumor cells in the bone marrow. So there's a great renewed interest in blocking the lysosome function autophagy with chloroquine derivatives. And so we do need to understand this better. And again, I'm not going to go through all of this, but really additional studies are warranted to fully understand the mechanism of chloroquine derivatives and more potent PPT1 inhibitors. And I want to close by saying, I think this analysis of comparing CRISPR knockout, siRNA, and chemical inhibition may be critical for many other targets in cancer. And we may find that there's a lot of differences when we do this for other drugs as well. So I want to thank the people that did the work in my lab. You can see Marianne Chrissy, Amanda Versace, Vaibhav Jain, and our new members, Arpana, Surabhi, Aaron, and Anna, and former member Monica Bardwaj as well. The chemical inhibitor work was done by Vito Rebecca and many others listed here. Our hydroxychloroquine trials are, I want to thank the UPenn Clinical Research Unit staff, the Tang lab for the proteomics, the Winkler lab for the chemistry, Dr. Zhu's lab for pathology, and Phyllis Gimity for biostats. Our funding comes from the NCI, as well as philanthropy with the Tara Miller Melanoma Foundation. And I want to thank the patients and their families for joining clinical trials, and I'm happy to take any questions that you have. Thank you.
Thank you, Ravi. The audience has submitted several questions, so let's get to them. So since there are clinical trials of novel PPT1 inhibitors for cancer ongoing, does your work suggest these agents may not be effective?
I'm sorry, say it one more time.
Of course. So there are now clinical trials of novel PPT1 inhibitors for cancer currently ongoing. So does your work suggest that these agents may not be effective?
No, I think that they are very toxic to cancer cells. And the question is PPT1 really the sole target, or are these, are the chloroquine derivatives actually impairing membranes in the lysosome or other mechanisms to cause that lysosomal lipid peroxidation? That's a subtlety, but it really suggests, methods to remove PPT1 may not be the right way to do it.
Sure. And could the conditional knockout mouse, you were saying about it could be used to study other lysosomal functions and other contexts. So can you describe a bit more about those contexts?
Yeah, so we got contacted by a neuroscientist at Harvard working on Alzheimer's disease, where protein clearance and proteostasis is a critical pathway of study, and having this tool of selectively blocking lysosomal function in his mind is very, very important. And so we were able to send him that mouse. And you could say the same thing for numerous other discoveries where the autophagy and proteasomal pathways are important, the lysosomal pathways are important. For instance, a recent paper showing that reinvigorated CD8 T cells after immune checkpoint are really upregulated for autophagy and lysosomal function. So what would happen if we use the PPT1 knockout mouse in a CD8 context, for instance?
Sure. And for your PPT1 inhibitors, you're not thinking that they'll be used for reducing cancer risk. You're thinking, of course, for like more therapeutic after someone has been diagnosed, correct?
That's the way that we have been. I have been operating for this whole time. I think the CLEVER trial, which I showed at the end of my talk, is really pointing out that if we move this earlier and really talk about preventing recurrence, that's what the CLEVER trial, the endpoint is preventing recurrence. it may be that a smaller volume disease is where the cells may be more autophagic and more sensitive to a PPT1 inhibitor. But it's hard. It's hard to do those studies in a registration format and have drug companies really focused on that. But we are engaging some partners to see if they're interested.
Wonderful. And so how do cells adapt to PPT1 inhibition, or how do you hypothesize they do?
Right. I honestly don't know. It seems to be the only thioesterase in the lysosome. Thioesterases are important because palmitylation is the only reversible fatty acid modification. So what is performing that thioesterase function in the lysosome? I don't know. It's not PPT2 because PPT2 doesn't have that enzymatic function. And so we've looked briefly at whether APT1 or APT2, the cytoplasmic thioesterases, can somehow be localized to the lysosome or endosomes, and maybe the endosomes take over and the cells are surviving on endosomal recycling degradation. But it's an open question. We have the data set to study this further, and it's published, so anybody can work on this.
Fantastic. So how does PPT1 interact and modulate the VATPase dependent selective autophagy?
Right. So components of the VATPase, which is multi-protein complex, are palmitoylated. And again, the reason why it's the only reversible fatty acid modification is we believe palmitylation is a signaling mechanism to localize proteins to where they should be. So when palmitoylated, the VATPase is on the plasma membrane. When depalmitylated, it's localized to the lysosome. It's stuck there in the lysosome. So the membrane between the plasma membrane and the lysosome is constantly being shared because of receptor recycling. So it comes in palmitoylated, and the PPT1 will depalmitylate it, and it'll stay in the lysosome.
Wonderful. And looking at your knockout, so your PPT1 FL whole body knockout, did you notice any phenotypes during embryonic development?
Just to be clear, we developed a conditional knockout. So the cells have complete PPT1 expression unless they're exposed to Cre recombinase. So there is a whole body PPT1 knockout developed by Sandy Blackburn many years ago. And again, those mice are fine, except they developed neurodegenerative disease because their neurons have huge amounts of lysosomal garbage that hasn't been cleared. And they die of basically seizures and lethargy and so forth. But not really, as far as I understand, any systemic problems, which is really surprising because, again, lysosomes will lack PPT1 in the whole body, liver, pancreas, and still those organs are okay. It's the brain that's selectively sensitive.
So how are the chloroquine trials in cancer going right now? And is there any activity thus far?
Yeah, there has been some activity. There in a randomized study in pancreatic cancer, actually two, one in the stage 4, one in the neoadjuvant setting. There was a significantly increased response rate when hydroxychloroquine was combined with chemotherapy. However, there was no difference in overall survival, which is the primary endpoint of those studies. Of course, they were small academic studies that were probably underpowered to detect a survival benefit. Similarly, in a melanoma study, that I ran, we saw a significant increase in response rate, meaning the tumor shrank more, but the duration of response was no different compared to historical controls. So that really speaks to resistance mechanisms, and our lab has found lipid metabolism is upregulated when you block lysosomal function, and probably saves the cell from dying and saves the cell from the lipid peroxidation that these drugs are producing. The breast cancer study I told you about is probably the most successful, with an 80% clearance of disseminated tumor cells with this 6-week treatment. That's a very compelling reason to continue this research, I believe.
So do you think that some of these effects could be cancer dependent, like dependent on which cancer you have or certain mechanisms? Like, will you see the same results, you think, if you look at melanoma versus breast cancer? It doesn't seem like it.
Yeah, I don't know the answer to that. We've looked for biomarkers. There is not a major difference in expression of autophagy genes. There is a difference in expression of PPT1 and other lysosomal genes. So there is some evidence that pancreatic cancer is kind of a go-to cancer for many reasons for autophagy studies. The difficulty is that pancreatic cancer has had very few hits as far as treatments that work over the years, and the patients are very sick. Very hard disease to develop new drugs unless they have major anti-tumor effects by themselves. And autophagy inhibitors may someday reach that threshold, but right now they're really combination plays. So it's very difficult to develop pancreatic cancer. But I think there's room to test this in many other cancers and continue to work on this field.
Fantastic. And are there any other autophagy inhibitors entering or in clinical trials for cancer?
Yes, there is a ULK1 inhibitor that is a highly selective and potent inhibitor from Decipher Pharmaceuticals that is in phase 1 clinical trials right now. I'm an investigator on it, so I can't really talk more about it.
Fair enough. Looks like that's it. So thank you, Ravi, for that discussion. We appreciate your presence here today.
Thank you.
If anyone has further questions, please consider reaching out to the speaker directly. His contact is now shown on the screen. So as a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. Make sure to follow the scientists on social media for updates on future webinars. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Noor Gamo and Ravi Amravati, as well as our sponsors, Taconic Biosciences, BioXcel, Aviva, and Syncell. Thanks, everyone, and goodbye.